Our next speaker is Dr. Tatler from Nottingham. Enhanced integrin B-beta 6 expression by transcriptional regulation of the B6 subunits.
Thank you. So good afternoon, everyone. I'd like to start by just thanking Luca and the rest of the organisers for asking me to come and present my data to you today. I'm a postdoc working at the University of Nottingham for Geezer Jenkins, and Geezer's group are predominantly interested in the role of the alpha-V beta-6 integrin in idiopathic pulmonary fibrosis. And I'm going to present some data to you today looking at how expression of this integrin is regulated and how it might be enhanced in IPF. So I'm not going to give you lots of background about IPF. We've heard lots about it today, but I just want to note that alpha-V beta-6 has a fundamental role in fibrogenesis. And this was originally highlighted in a paper in 1999 by Dean Shepherd's group, who showed that mice that were knocked out for this integrin were protected from bleomycin-induced pulmonary fibrosis. These same mice were later shown to also be protected from radiation-induced pulmonary fibrosis. So together these studies highlighted alpha-V beta-6 as a potentially important therapeutic target, and a monoclonal antibody was developed. This antibody has since been shown to inhibit both bleomycin-induced pulmonary fibrosis on the left and also radiation-induced pulmonary fibrosis on the right. And this antibody has now been humanized and is in phase 2 clinical trials, and I think we'll hear some more about that from Sheila Violet later in the conference. So how is alpha-V beta-6 involved? Well, the original paper neatly demonstrated that alpha-V beta-6 binds to and activates the latent TGF-beta complex. And TGF-beta is a potently pro-fibrotic cytokine that's thought to be the central mediator in IPF. Now, our group and others have shown that alpha-V beta-6 activates TGF-beta in response to a wide variety of signals that are generally released during the response to injury. And there's just some data here showing that alpha-V beta-6 activates TGF-beta in response to lysophosphatidic acid and also thrombin. So in normal healthy tissues, expression of alpha-V beta-6 is kept very, very low, and so low in fact that it's often barely detectable by immunohistochemistry. However, in response to injury, its expression is dramatically upregulated, and this has been well documented in several papers looking at its expression in IPF. We've recently looked at the mRNA levels of specifically the beta-6 subunit of the integrin and shown that the mRNA levels of the beta-6 subunit are increased in fibrotic tissue from patients with IPF compared with non-fibrotic controls. So what do we know and what don't we know? We know that alpha-V beta-6 is upregulated in IPF and also in vivo models of pulmonary fibrosis, but we don't really know how. We don't really know whether the molecular regulation of alpha-V beta-6 is dysregulated in IPF, and we also don't know what the functional significance of this increased expression is. So I started these studies looking at the effects of TGF-beta on alpha-V beta-6 expression. I stimulated lung epithelial cells with TGF-beta and assessed alpha-V beta-6 cell surface expression by flow cytometry, seen at the top, and found that alpha-V beta-6 expression increases in a dose-dependent manner in response to TGF-beta. When I looked at mRNA levels of specifically the beta-6 subunit, TGF-beta increases mRNA in a time-dependent manner. I've also kindly been given an integrin beta-6 promoter reporter that drives a luciferase gene. When I transfect this reporter into the lung epithelial cells and stimulate them with TGF-beta, we see an increase in beta-6 promoter activity. So together, these data suggest that TGF-beta increases alpha-V beta-6 via transcriptional upregulation of the beta-6 gene. So I wanted to then look in further detail at the promoter and highlight which region of the promoter could be important for regulating this response. So I took the original promoter construct that I used in the previous slide, and from it created a series of deletion mutants. As the mutants get progressively smaller, transcription factor binding sites are lost from the promoter, and this allows us to investigate specific regions of the promoter. When these constructs were transfected into the lung epithelial cells and stimulated with TGF-beta, I found that the full-length promoter responded to TGF-beta as we saw before, as did the first mutant, the -818 mutant. However, loss of a cluster of transcription factor binding sites that included a single SMAD binding site from the promoter resulted in an inability to respond to TGF-beta stimulation. So just to look at this SMAD site in further detail, I've used site-directed mutagenesis. I've mutated each of the 3 most distal SMAD binding sites in the promoter construct that I've been using individually, and then transfected these constructs into the lung epithelial cells and stimulated with TGF-beta. As shown before, the full-length promoter responded to TGF-beta, as did constructs with a mutation in SMAD site 5 and SMAD site 4. However, mutation of SMAD site 3 resulted in a lack of response to TGF-beta, suggesting that this is the SMAD site that's mediating the increase in promoter activity when we stimulate with TGF-beta. So to assess whether SMAD proteins bind directly to the beta-6 promoter, I've used a technique called Chromatin Immunoprecipitation. And for the non-CHIP people in the audience, I'll just give you a quick overview. We can cross-link proteins such as transcription factors to DNA by fixation. We can then add an antibody directed to the protein of choice and protein G magnetic beads, which allows us to pull down the protein that we're interested in, and with it, it will bring any DNA that it's bound to. We can then do PCR analysis of this DNA to see which gene promoters have bound to which transcription factors. So using this technique, we've been able to demonstrate that in response to TGF-beta, both SMAD3 and SMAD4 bind directly to the integrin beta-6 promoter in the region that we've previously highlighted as being important. We've also assessed binding of SMAD2 to this region, but we get very unreliable results and we don't get any reproducible binding of SMAD2 to the beta-6 promoter in response to TGF-beta. So to look at the functional consequences of SMAD3 in further detail, we were kindly given some dominant negative constructs directed towards both SMAD2 and SMAD3. When I co-transfected these constructs into the lung epithelial cells with the beta-6 promoter reporter, I found that dominant negative SMAD2 had a marginal effect on the promoter activity. However, dominant negative SMAD3 inhibited both basal and also TGF-beta-induced promoter activity, highlighting a role for SMAD3 rather than SMAD2. I've also used these constructs to look at their effect on cell surface expression of alpha V beta-6. TGF-beta, shown in the blue line in these flow diagrams, is able to increase alpha V beta-6 expression in cells that hadn't been transfected, in cells that transfected with an empty vector control, and also cells that are transfected with dominant negative SMAD2. However, this response is blunted in cells that are transfected with dominant negative SMAD3. We've also gone on to do some in vivo work to look at whether this applies in vivo. And in collaboration with Jack Goldie and Martin Kolb, we've used the SMAD3 knockout mice and the adenoviral TGF-beta model of pulmonary fibrosis. So when wild-type mice are treated with adenoviral active TGF-beta, they develop widespread pulmonary fibrosis. And we also see a dramatic increase in alpha V beta-6 expression within the alveolar epithelium. However, when SMAD3 knockout mice are treated in exactly the same manner, they don't develop pulmonary fibrosis, and we also don't see such a dramatic increase in alpha V beta-6 within the alveolar epithelium. So while we were investigating a role for SMAD3 in mediating TGF-beta-induced beta-6, we came across quite an exciting observation. So if I can cast your minds back to this figure, when we lost the cluster of transcription factor binding sites between the -818 and the -731 mutant, we got a dramatic increase in basal activity of the promoter, suggesting that there might be a loss of one or more transcriptional repressors within this region. So we just looked at this region in further detail and did sequence analysis just to find out which transcription factor binding sites were in that region. And we found quite an array of transcription factor binding sites that could be responsible for repressing the integrin beta-6 gene basally. Now, we've actually investigated 3 of these now. We've done work on the glucocorticoid receptor using dexamethasone, the progesterone receptor using progesterone, but I'm just going to present some data to you today where we've looked at ELK1. So ALK1 is a nuclear transcription factor that's capable of both activating and repressing gene transcription. And because it's a nuclear transcription factor, we've had to use an siRNA approach. So I used siRNA directed towards ALK1 to knock down expression of ALK1 and then transfected the cells with the beta-6 promoter. And we see both an increase in basal activity of the promoter and also an increase in the response to TGF-beta. When we look at alpha-V beta-6 cell surface expression 3 days following transfection with the siRNA, we find that the ALK1 siRNA causes an increase in alpha-V beta-6 expression compared with the scrambled siRNA control. And finally, we've been doing some chip analysis on patient tissue from patients with IPF. And we've come across some quite interesting discoveries. So when we looked at SMAD3 binding to the beta-6 promoter, we found that in a small subset of patients, there is increased binding of SMAD3 to the beta-6 promoter. And perhaps even more excitingly, we've been able to demonstrate that in some patients with IPF, there is decreased binding of the transcriptional repressor in IPF. And as Toby highlighted earlier this morning, there is quite vast heterogeneity within IPF, and this is just shown in these figures. So just to give you a quick overview of what we've found, TGF-beta on the cell surface is activated by the alpha-V beta... Oh!
Help!
Help! Okay, sorry. So alpha-V beta-6 is expressed on the cell surface, bound to the latent TGF-beta binding complex. It can activate TGF-beta, which binds to its cell surface receptor, phosphorylating SMAD3, which translocates to the nucleus. Here, it activates transcription of the beta-6 gene, leading to increased expression of alpha-V beta-6 on the cell surface. However, in IPF, at least in some patients, there could be increased binding of SMAD3 plus decreased binding of ALK1, leading to more beta-6 transcription and even more alpha-V beta-6 on the cell surface. So what do we know now? Well, we know that TGF-beta increases beta-6 expression and that this leads to alpha-V beta-6 expression increasing. We know that this is due to SMAD3 binding to the promoter at a very specific single SMAD binding site. And we've also found that ALK1, a nuclear transcription factor, can repress basal activity of the gene. We've also found that in some patients with IPF, there is increased binding of SMAD3 to the beta-6 promoter, which may lead to increased expression. And we've also found that some patients with IPF have decreased binding of a transcriptional repressor, which could also lead to enhanced alpha-V beta-6 expression. But what is the functional significance? Well, I urge you to go and see 2 posters by my colleagues, Alison John and Gauri Saini, who will hopefully be able to answer some of those questions for you. So I'd just like to thank everyone in Giza's group and also our various collaborators without whom the work wouldn't be possible at all. And also I'd like to invite you all to come to Nottingham next summer, to a conference Giza and I are organising on behalf of the British Association for Lung Research, Curing Pulmonary Fibrosis, the Holy Grail. Thank you very much.
Dr. Tatler's very elegant series of experiments are open for discussion.
Thank you.
So let me ask just a quick question.
Okay.
Do you think in your very elegant study you've demonstrated the paracrine cycle that allows IPF to continuously generate even without exogenous injury?
So I don't, I've not been able to show you all the data. We have been able to show that blocking the alpha-V beta-6 integrin or TGF-beta leads to a decrease. So there is a basal level of TGF-beta activation that regulates the basal activity, the basal expression of the integrin. However, what we've not been able to demonstrate yet, and what I think is the most important aspect, is how this this kind of a cycle becomes out of control in IPF. Like clearly the epithelial cells are able to, in normal conditions, regulate their expression of the integrin via TGF-beta, but clearly something goes wrong in IPF and it just, right. I mean, it may be due to ALK1, but we would need a much bigger cohort of patients and tissue to be able to assess
but it would seem like a bad idea to be upregulating the integrin, which upregulates TGF-beta, and then you get the cycle, right? I mean,
Sure,
yeah,
sure.
Okay.
Dr. Shepard?
Thank you.
One piece that still seems like there's something missing is what really is regulating this repressor, because it looked like there was a maybe 30 or 50-fold increase in expression when you get rid of the repressor, and a maybe 2 or 3-fold of that is explained by L1.
Sure.
So do you have any good ideas about what else might be regulating the repressor
Sure.
in addition to L1?
So I think it could be any one of the other transcription factor binding sites that we've shown. We've also done some work with dexamethasone, and our data suggests that treatment of epithelial cells with dexamethasone inhibits alpha V beta 6 expression. Now, whether this is due to the glucocorticoid receptor repressing the gene, or whether it's due to the glucocorticoid receptor interrupting SMAD signaling prior to nuclear events, which it has been shown to do, we're not sure yet, but it looks as though the dexamethasone could inhibit alpha V beta 6 expression as well.
I guess one other comment is the data from analysis genome-wide of regions that activate and repress genes suggests that many of the important regions are much further than 1,000 nucleotides away.
Yeah.
So you have some thoughts about how to approach
Certainly,
the distant regions that might be regulating
Certainly,
expression
I
in
mean,
vivo?
we haven't looked that far. The promoter reporter that we've been using is only 1.1 kb. And certainly I was quite surprised to see this presence of the repressor as close as we found it, because like you say, the published data would suggest that it's being repressed much further up, which may also be the case as well, but this data has only looked at quite close.
Thanks.
Thank you.
Any further questions? Thank you very much. Oh, you have, yes.
So do you have a handle on what the co-activator would be with SMAD3? Because SMAD3 on its own isn't enough, obviously, to get the transcriptional machinery going. Do you have any thoughts on what the co-activator might be? And do you think that the co-activator and the repressor pathways are competing?
I'm not sure, but I think I think that's where this is going next. I mean, certainly we've shown that SMAD4 will translocate, but whether it's translocating with SMAD3, because the time course is quite different. So yeah, I don't really have an idea of what the co-activator could be, but I think it could be quite exciting to see exactly what's going on with this integrin.
And can I ask you another question just about the approach? Because obviously these sites are now all nicely available in these short promoter constructs, but obviously the gene in its endogenous environment is wrapped around heterochromatin, etc. How are we going to tease out as a community whether these sites are actually available and therefore important when we're using these slightly artificial systems? Do you have any thoughts?
One of my thoughts about the ELK1 data is precisely that. So, I mean, we haven't looked at expression levels of ELK1, but based on the literature and based on studies that we've done in other diseases like asthma with other transcription factors, I would hypothesize that the expression levels between IPF and non-fibrotic controls are actually the same. And what I think could be at play are epigenetic changes in the DNA that's perhaps not allowing ELK1 in some patients with IPF to bind to the beta-6 promoter, and therefore it can't repress it, would be what I would hypothesize.
Great, thank you.
Thank you.
Okay, thank you very much.
Okay, thank you.